Hello, physiologists. I want to talk to you today about tubular reabsorption and secretion that occurs down the length of the renal tubule in the nephron. Reabsorption is where you're going to return most of what you filtered out of the glomerulus back into the bloodstream. About 65% of the filtrate is going to be reabsorbed, and nearly all of the filtered water is going to be reabsorbed, which means we'll return it back to the blood. Most of this process will take place in the proximal convoluted tubule, and the reason for this is there is a structural modification that helps reabsorption, and that is the presence of microvilli. So these are extensions of the plasma membrane that increase surface area, and as we'll see soon, you have a lot of transporters, you have a lot of transport that takes place along the membrane, and so those microvilli are going to help with the reabsorption. Secretion is movement in the other direction. This is where you're moving material from the blood into the tubular fluid. And so in particular for substances that are not filtered very well, secretion is an important method to help get products that you don't want anymore out of the blood and into the tubular fluid. And remember, at the end of the nephron, whatever has not been reabsorbed will be excreted into your urine. Both of these processes occur simultaneously along the entire length of the renal tubule, but a lot of times when we're discussing these processes, it's easier to just talk about them one at a time. But please know that they are both occurring simultaneously down the length of the tubule. Here is a quick overview and comparing two different primary routes, transcellular and paracellular. So transcellular routes are where substances are transported from the tubular fluid here through the cell, and frequently this is going to involve transporters along the cell, and then through the basolateral side of the cell and then into the blood. Remember that ions, large substances, and other things are going to have difficulty diffusing across a plasma membrane. So we are going to be using quite a few transporters in order to help move those substances across either the apical side of the membrane or the basolateral side of the membrane. Some substances are also taken between cells, and this is the paracellular route. And they go between the cells to go to the bloodstream that way. And this is often what happens with solvent drag. And so as you have a lot of water that is leaving, it tends to kind of drag some of these other substances along. But a lot of the times we're going to be focusing and learning more about the transcellular route. But you can go between the cells as well. In order to cross to the bloodstream, you're going to need to cross through the simple epithelium of the tubule wall. You have simple cuboidal epithelium. If you go through that transcellular route, you do have to cross two different cell membranes, and so you may see two different transport proteins that are helping to move a substance across from the lumen of the tubule all the way to the bloodstream. Peritubular capillaries will have a very low hydrostatic pressure. Remember, these capillaries receive their blood from the efferent arteriole of the glomerulus, where you've just filtered out a lot of material, and so there's not very much pressure pushing against the walls of these capillaries. So very low hydrostatic pressure, and this helps to facilitate reabsorption. It's very easy for material to move into these capillaries. In the tubule, there are some specialized cells that respond to specific circumstances, specific hormones. Principal cells have receptors for aldosterone and antidiuretic hormone. Intercalated cells, you have type A and type B intercalated cells. These help to regulate the pH of the urine and the blood. Type A cells will eliminate acid, and type B intercalated cells will eliminate base. So that's an easy way to remember them. These cells are found along the distal convoluted tubule, the collecting tubules, and collecting ducts. So we'll talk about those a little bit later. With all of these transporters, there is a maximum amount that you can move across a tubule given a given period of time. And this is because there's only so fast that those transporters can go, and there's only so many transporters that you have. And so we'll talk in just a second about glucoseuria, where your glucose transporters are overwhelmed. So you have a specific amount of a substance that you can transport in a particular period of time. Renal threshold is the maximum plasma concentration of a substance without it appearing in the urine. So this is basically how much can your blood hold of a specific substance without any of it being secreted or not reabsorbed and it appearing in the urine. Here is glucoseuria, so it's an example of where you're overwhelming your glucose transporters. Normally you want to reabsorb all of your glucose. Glucose is a very good energy substance. If you have excess, you put that into storage. You'd never ever get rid of glucose. So you can see in normal glycemia, here's your blood. All of these little dots are your glucose molecules. You filter a lot of that out and you head down the renal tubule. Here are your glucose transporters and you end up with all of that glucose returning back to your blood. And by the time you get down to this later tubule, you don't have any glucose left because it has all been reabsorbed. And so when you look at a urine sample, a urinalysis, it is glucose free. However, in hyperglycemia, you can see that there's lots and lots and lots of glucose here. Remember, especially for diabetes mellitus, your insulin is not working properly or you're not producing enough of it, so you have a lot of glucose in your blood. This is way too much glucose for your transporters to handle. They can only work so fast and you only have so many transporters. By the time you get to the end of the tubule, you still have glucose that has not been reabsorbed. And so you end up excreting some of that glucose into your urine. And so this is one big way that you can use to diagnose diabetes mellitus. If you have that glucose appearing in your blood, you get diagnose that hyperglycemia. Here's how glucose reabsorption looks in the renal tubule. All of these microvilli show me that this is a proximal convoluted tubule cell, and because distal convoluted tubules do not have these microvilli, they're just flat-faced cells. So this should look very familiar if you have learned about carbohydrate absorption in the small intestine. We're going to start in a very similar way. So I'm actually going to start on the basolateral side of the cell that's closest to the blood, the peritubular capillary. You have a sodium-potassium pump here, and of course you're pumping potassium in, but I want to focus on moving the sodium out of the cell. So reabsorption of sodium is very important, just in general, because it helps to control water in the body as well. So, but as you're moving the sodium out, you are creating a concentration gradient. Low levels of sodium inside the cell and higher levels are in the tubule because you've just filtered all of this material out, including sodium, including glucose. And so you have a high levels of those in the lumen of your tubule and low levels that are being created in the cell thanks to the sodium-potassium pump. Here's your sodium glucose transporter right here, same one that we see in the small intestine for absorption. So as sodium moves down its concentration gradient, it drags along glucose for the ride. And then glucose can leave through this facilitated diffusion transporter here, and then glucose ends up in the blood in addition to sodium. So in this way, as you're reabsorbing glucose, you're also going to be reabsorbing sodium. Sodium reabsorption is very highly regulated. It's very important because it regulates other solutes and it also regulates water. And you do have some control over how much sodium you reabsorbed by hormones, aldosterone. For example, aldosterone will increase how much sodium you reabsorb from your tubule. And again, they establish that concentration gradient of low concentration of sodium within the cells so that it is easier for sodium to move through the cell. Here's another picture of what sodium reabsorption looks like. On the left side, this is a picture of a proximal convoluted tubule cell, and it's a little bit sideways compared to the picture that we looked at before, but here's your lumen at the bottom. Here are the microvilli and your proximal convoluted tubule cell here, and then here's the basolateral membrane up here, and then you have your peritubular capillary up here. So most of your sodium is going to be reabsorbed the same way that we discussed before when we were talking about glucose. You have a sodium-potassium pump that, multiple sodium-potassium pumps, just one pictured. But here's one in the basolateral membrane. It's constantly moving potassium into the cell and sodium out of the cell. And that sodium that's moved out of the cell on the basolateral side moves into your bloodstream. You have established a low concentration of sodium inside the cell, and so thanks to this gradient that's created, you have more amounts of sodium in the tubule that are able to be moved into the cell through this facilitated diffusion transporter. And so sodium is able to move through this transporter, through the cell, and then through the sodium-potassium pump, and then be reabsorbed into the blood. If this is not enough sodium reabsorption, you can have aldosterone secreted. And so this picture on the right is a different area of the cell. And this is a cell in the distal convoluted tubule, the collecting tubule, or the collecting duct. So this is very late in the nephron or just past the nephron. Here's an aldosterone receptor right here in the green. When you have aldosterone bind to its receptor in these principal cells, remember there are specialized cells that have receptors for ADH or for aldosterone. So the way that aldosterone works in these cells is you have sodium-potassium pumps. So you have more sodium-potassium pumps, so you're continuing to pump more and more sodium into the cell. You're also going to create more sodium channels and more aquaporins. Aquaporins are specialized channels for water that help you to reabsorb more water through osmosis. So as you're moving more and more sodium, thanks to these extra transporters, they move through the cell and they are shuffled out of the cell thanks to more sodium-potassium pumps. You're absorbing more water, and then thanks to the aquaporins, more of that water is moving into the blood. This picture is also showing some more sodium reabsorption, but it's coupled with hydrogen ion secretion. Hydrogen ions are not filtered very well out of the glomerulus, and so you really need some methods of getting rid of hydrogen ions out of the blood to keep the blood from becoming too acidic. And this picture looks complicated, but I want you to focus on this middle part right here. This equation hopefully looks familiar to you because we see it in the respiratory system, we see it in the digestive system, and this is a very common equation. So we're going to focus on this middle part right here. You have carbon dioxide, which is prevalent. It's a metabolic waste product, combines with water, which is also very prevalent in the body. These proximal convoluted tubule cells have an enzyme called carbonic anhydrase, and this enzyme is going to help speed this reaction up a little bit. So carbon dioxide plus water combine to make carbonic acid, this H2CO3, and then carbonic acid dissociates into hydrogen ion and bicarbonate ion. Bicarbonate ion is a really great buffer. It's very useful in the body. We want to keep it. So bicarbonate ion moves into the blood. This is facilitated diffusion transporter right here. So you're moving this bicarbonate into the blood, and this hydrogen ion is going to move into the tubule lumen. And so you're getting rid of this hydrogen ion. It is being secreted into the tubule lumen. And you may notice that with this transporter, hydrogen ion is not the only one moving. We're also moving sodium. This is an antiporter. As sodium moves down its concentration gradient into the cell, hydrogen is moving out of the cell. Since they move opposite directions, it's an antiporter. And hopefully you're wondering, if sodium is moving down its concentration gradient, do we have another one of those sodium-potassium pumps helping to make sure that you maintain a low concentration of sodium inside the cell? And yes, we do. Here's your sodium-potassium pump on the basolateral membrane, continuing to move sodium out of the cell so that sodium from the tubule can go down its concentration gradient, and as you use that potential energy, the hydrogen ion can be pushed into the tubule lumen. So this is how we're going to get rid of hydrogen ions, reabsorb some more sodium, and also reabsorb some bicarbonate. Bicarbonate is freely filtered, but you don't filter that much hydrogen. So again, you want to reclaim the bicarbonate buffer, which is a weak base, and you want to get rid of, you want to secrete the hydrogen ions, because this is going to help maintain your blood pH homeostasis. Here's another picture showing bicarbonate reabsorption. Again, we have this picture right here. So the right side of this picture is the same one that we saw before. Carbon dioxide plus water creates carbonic acid. Carbonic acid dissociates into hydrogen ion and bicarbonate. We reabsorb bicarbonate through facilitated diffusion. we do have our sodium-potassium pump over here, maintaining a low concentration of sodium. Sodium comes down its concentration gradient from the tubule into the cell, and then that sodium will just get shuffled out through this sodium-potassium pump. As sodium moves down its concentration gradient on the apical side of the membrane, it pushes out this hydrogen ion. Now this hydrogen ion can combine with bicarbonate that's in the tubule and then create carbonic acid. So you see the same equation, but we're moving backwards here. And then as this carbonic acid dissociates, you get water, and then you have CO2. So you can kind of have this recycling of the CO2 between the tubulumen and the bicarbonate. And by turning this hydrogen ion, you don't want your urine to be incredibly acidic either. Basically, this hydrogen ion is ultimately going to be converted and turned into water, which is a much more neutral compound. So you don't have all of these hydrogen ions that are floating in your tubulumen. You can help buffer what's in your tubule in addition to buffering what's in your blood through the reabsorption of bicarbonate. Here is what's occurring in your proximal convoluted tubule cells compared to a type A intercalated cell, which is located later in the tubule. So this picture on the left is looking at the same things that we were looking at before. You see our equation occurring in the tubular fluid. Bicarbonate plus hydrogen ion makes carbonic acid, which dissociates into carbon dioxide and water. Carbon dioxide diffuses into the cell, combines with water, turns into carbonic acid. You want to move out the hydrogen ions here, and then save that bicarbonate and move it into your blood. Type A intercalated cells are only found in the late distal convoluted tubules and collecting tubules and collecting ducts. But they work much the same way, they just don't require the transporters. So you have carbon dioxide, we move through our equation, carbonic acid dissociates, you save the bicarbonate, move it into the blood. Remember, type A eliminates acid. So this acid, this hydrogen ion, is moved into the tubular fluid so that you can secrete it. You want to get that out of your body. Water movement occurs by osmosis, either through paracellular transport, so that's in between the cells, or through aquaporins. You do have obligatory water reabsorption that occurs, which is basically water follows salt. So whenever you're moving sodium, you're going to move water as well. Water is attracted to that salt. Water will follow that salt. And so anything that's going to affect sodium reabsorption will also affect water reabsorption. So aldosterone increases how much sodium you reabsorb. You automatically are going to increase how much water you reabsorb. ANP, atrial natriuretic peptide, decreases how much sodium you reabsorb. And so therefore you're going to decrease how much water you reabsorb because you'll have a smaller amount of water following that smaller amount of salt. ADH, antidiuretic hormone, increases your water reabsorption. It is not tied to the sodium regulating hormone, so ADH can function independently of other hormones like aldosterone. ADH produces a smaller amount of concentrated urine because you're pulling more of that water out of what would have been in your urine and returning that to the blood. If you're dehydrated, this is one instance where you would have more ADH being secreted. And here's how that works. So here's, again, in your principal cell. Principal cells are the ones with the receptors for ADH and aldosterone. This is only going to be found in the distal convoluted tubule, the collecting tubule, or the collecting duct. So here's ADH, antidiuretic hormone that's bound to its receptor. And you have additional aquaporins that have been inserted into the membrane. And so more water is able to flow through the cell and then move into the blood. Now, even though there's not sodium that is being affected here, later on when we talk about countercurrent mechanisms and the nephron loop, there are some other circumstances that are present in the nephron that enable you to absorb more and more and more water. But that is a topic for another time. Along the length of the nephron loop, you have some specialized symporters. And I say down the length, but this is really only found in the ascending limb of the nephron loop. So here's our picture of our nephron right here. Here's your proximal convoluted tubule. You have the descending limb and the ascending limb. And the ascending limb is specifically where you'll find these transporters. So these symporters here move sodium, 2-chloride, and potassium, and all three of these substances move into the cell. Notice we're moving 2-chloride and just one of the others. Sodium will be reabsorbed thanks to this sodium-potassium pump. So sodium moves out of the cell and into this blood vessel. Remember, since we are along the nephron loop, the blood vessels are called the vasa recta instead of the peritubular capillaries. The potassium is recycled out of the cell, and then these 2-chloride will leave through these leakage channels here to move into the vasa recta. So when we look at what's in the vasa recta, we have 2 negatively charged ions, 2-chloride, and only 1 sodium ion, which is positively charged. So you're moving more negatively charged ions than positively charged ions into the blood. This is going to attract other cations, other positively charged ions, which will help balance that charge. And so while this symporter strictly just moves sodium, chloride, and potassium, you're also going to end up reabsorbing other cations, additional sodium, additional potassium, calcium, and magnesium, which will be moved through the paracellular route into the blood. For potassium reabsorption, a lot of the potassium will be reabsorbed in the proximal convoluted tubule here. You do get some of that potassium reabsorbed in the ascending limb of the nephron loop, thanks to those sodium, chloride, potassium symporters that are present there. And maybe even if the potassium isn't strictly reabsorbed through transporters, remember that you're going to get some of that potassium that's attracted to the extra chloride in that limb. You have regulated potassium reabsorption along the collecting tubules. Type A cells that are present in the collecting ducts and collecting tubules will constantly reabsorb potassium, and then the principal cells can adjust your potassium that's secreted depending on whether aldosterone is being secreted or not. Remember, as aldosterone reabsorbs more sodium and more water, potassium is secreted at the same time. And that's partially due to those sodium-potassium pumps moving potassium the other way, and then you've got potassium leakage channels that helps it to be secreted. So you have regulated reabsorption and secretion of potassium throughout the nephron. Calcium and phosphate are also regulated, and this is done through parathyroid hormone. In the early distal convoluted tubule, there are receptors for parathyroid hormone, and this hormone will cause those cells to inhibit phosphate reabsorption, and so you're not going to move as much phosphate back into the blood, while you increase calcium reabsorption. And so you're going to move more calcium back into the blood, but you're not going to move as much phosphate. And the reason for this is because calcium connects with phosphate, it binds to phosphate, and then that calcium phosphate gets stored in your bones. If parathyroid hormone is secreted, that means you have low plasma calcium. And so you don't want that extra calcium that you're reabsorbing to just move straight to your bones. You want it to stay in your plasma. And so by inhibiting phosphate reabsorption, you're making sure that it's just calcium that's moving to your plasma and it stays in your plasma, instead of combining with that phosphate and moving to your bones. So parathyroid hormone will regulate both phosphate reabsorption and calcium reabsorption. This is a recap. It's a very busy picture, but it does sort of show kind of a summary of reabsorption in the proximal convoluted tubule. So you can see some of the different types of receptors that are present along the basolateral membrane. See your sodium-potassium pump here. You see some potassium channels. Here's an aquaporin, the water channel that helps move these substances into the blood. And then you can see your sodium symporters, glucose, amino acids, some vitamins. They're all going to use this sodium symporter. As sodium moves down its gradient, it moves another substance. Here's an aquaporin on the apical side. Lipid-soluble substances can transport themselves through the plasma membrane without any problem. So you just have straight diffusion, no transporter needed. Remember, lipid-soluble substances normally don't have a problem moving through plasma membranes because the plasma membrane is a lipid bilayer. And so you don't need special transporters for those lipids. And then other ions in urea are able to move through the paracellular route. And so this is kind of a summary of different ways that you can move substances from the tubule into the blood, reabsorption. Some substances must be eliminated as waste, and so you have multiple different nitrogenous waste products, urea, uric acid, creatinine. These are all examples. You can see their structures here. Notice how much nitrogen is present in these structures, so this is why they're called nitrogenous wastes. You also have various drugs and bioactive substances whenever you take antibiotics. If you take other drugs such as morphine or marijuana, even just metabolic waste products like bilirubin, some metabolites from hormone breakdown, some hormones, whole hormones themselves, you don't want them floating around in your blood forever. And so you're going to eliminate these substances as waste. You're going to actively secrete these substances and make sure that they end up in your renal tubule and excrete these as in your urine. Here's a quick summary of tubular reabsorption and secretion. All of the substances in the blue boxes are being reabsorbed. So you can see, in addition to the arrows, you can see where these substances, what part of the tubule they're being moved to the blood. So all of these substances are reabsorbed at the proximal convoluted tubule. You can reabsorb these substances additionally in the distal convoluted tubule. Here's that symporter we talked about in the ascending nephron loop, showing reabsorption of these materials. And then in the pink boxes, these are the substances that are being secreted and in what parts of the nephron. So these substances, notice your nitrogenous waste products along with some drugs. These are secreted into the proximal convoluted tubule here. You have some more secretion of these substances in the collecting tubule and collecting duct here. So this is just a quick summary of what substances are reabsorbed versus secreted and where that process occurs along the nephron.